everyone, today I've got some really quick news for you about a study that was recently published in the journal Proceedings of the National Academy of Sciences. The research looked at various types of venomous centipedes, which are known to possess extremely lethal chemicals that get injected into the victim's bloodstream with a penetrating bite. The toxins can kill lizards and large insects, which are staples in the diets of these venomous carnivorous centipedes. But the toxins are so powerful that they can kill larger animals, like birds, mini mammals, and in a high enough dose, even humans. It's known that this toxin causes serious, often lethal problems with the lungs, with the heart beating, and with the neural function of the victim. But until now, the culprit chemical and the mechanism of action were a mystery. No one knew exactly what it was or how it worked. The researchers studied the golden-head centipede Scolopendra subspinipes mutilans, which is native to Eastern Asia. Now first, in laboratory conditions, they observed the centipede bite a mouse. Even though the mouse was nearly 15 times the size of the centipede, its toxin was strong enough to kill the mouse within 30 seconds. They extracted a sample of the toxins from the centipede, and they began to study them to identify the chemical components. The majority of the chemicals that they identified were relatively harmless or benign. They certainly didn't have the reactive capacity to kill the mouse. But one chemical in the centipede venom was quite different. The researchers identified the active ingredient, a peptide toxin called SSTX, which is short for the SSM spooky toxin. Apparently, this peptide toxin is able to disrupt the proper activity of potassium channels in the KCNQ family. These potassium channels are present in nerve cells, where they work to help manage and conduct action potentials and thus carry nerve signals throughout the body. When an organism is infected with SSTX from a centipede bite, the toxin is then able to shut down all of these KCNQ potassium channels, which then rapidly and massively disrupts potassium exchanges and ruins the functioning of the nerves. The heart will begin to beat irregularly, as the signals that coordinate its rhythmic beating are being shut off, they're being tampered with by this toxin. The lungs will struggle to expand, as the deep brain signals are being impaired. Muscles will twitch out of control, and the nerves report to the brain a terrible phantom sensation of pain. This leads to a relatively rapid onset of death, as multiple critical systems fail within the victim's body. Evolutionarily, this makes sense, as the researchers point out that producing this toxin is metabolically expensive. The centipede thus needs something that can be maximally effective because of the heavy energy investment, and a toxin that targets a super ubiquitous ion channel in the nervous system, like the KCNQ potassium channels, it's like a guaranteed payoff. It's a guaranteed kill. Now another cool part was that this research wasn't just about the identification of the toxin. The researchers also identified a compound called retigabine, which is able to reopen the potassium channels, and it thus works as an antidote or an antivenom. Studies like this are really exciting and they're really important because they help us learn more about the dangers that surround us in the natural world. This study goes above and beyond the norm by also identifying a potentially life-saving chemical antidote.